This method can help answer key questions in the RNA biology field about the mechanism and function of polarized RNA localization. The main advantage of this technique is that it allows for the real-time visualization of endogenous RNA trafficking in live tissues.
The implications of this technique extend toward a therapy of RNA-related diseases, because it has the potential to uncover new therapeutic targets.
Though this method can provide insight into RNA processes in the fruit fly egg chamber, it can also be applied to other systems, such as in zebrafish or culture cells.
Generally, individuals new to this method will struggle because of the challenges associated with the design and delivery of the molecular beacon probes.
For molecular beacon microinjection, select the 40x oil objective and mount a cover slip with a dissected egg chamber onto the microscope stage. Locate an egg chamber at the mid to late developmental stage that is properly oriented for microinjection. Then set up a microinjector with the appropriate injection and compensation pressures. Using the micromanipulator joystick, gently lower a needle loaded with 1 microliter of molecular beacon solution into the oil drop. Bring the tip into focus toward the periphery of the field of view. Perform a clean function to remove the air from the needle tip and to ensure that there is flow from the needle, and bring the needle to the home position. Focus on the egg chamber to be microinjected, and bring the needle back into focus near the edge of the egg chamber. Perform a fine adjustment of the Z position of the objective such that the membrane separating the follicle cells from the nurse cells is in focus. Insert the needle into a nurse cell for injection of the molecular beacons over a